Hello everyone. So this is regarding the lab diagnosis of cholera, and this is a brief summary regarding the lab diagnosis of cholera. So we will start the diagnosis by collection of stool, and this stool should be a fresh sample, and it should not be pre-treated, and the patient should not be pre-treated with any of the antibiotic. Now, this collection of stool sample can be done via the rubber catheter, which is a soft type and is lubricated with paraffin wax or any other lubricant, and its size ranges from number 26 to 28, and it is inserted 4 to 5 centimeter within the rectum, and then it is taken out, and the sample is stored in VR medium. Another way is through the rectal swab, which is already pre-dipped in the holding medium, that is Venkatraman medium or the alkaline peptone water medium. Okay, so these are the two important methods, the rubber catheter or the rectal swab. But another method where both of these are not available, means the holding medium is not available, is that we do cotton tip rectal swab in a sterile bag, and then after collecting the sample of the stool, we do also collect the sample of water of the suspected area and the food sample which is suspected, and then this all is transported through McCartney bottles that are around 30 ml, and these bottles contain alkaline peptone water or VR medium. Especially, mind you, in the case the sample is taken through rectal swab, we do collect the sample in alkaline peptone water or Carrie Blair containing McCartney bottles. And the sample is around 1 gram or 1 ml in 10 ml of the respective alkaline peptone water or VR medium. Okay, so after this collection, we examine the sample. Okay, so we can directly go for dark field illumination, and doing this directly gives a good result. In around 80% of cases, we can directly diagnose the disease within hours. But in cases where this is not diagnosed, we need to do the enrichment through culture. So when we do dark field illumination, we see uh, characteristic sign of shooting stars in dark sky. Okay, as you would have studied, as you would have seen the images of this. So this shooting star appearance with a darting motility is hampered when we give the anti-cholera serum, and this makes our diagnosis more definitive. In the case we don't get the result directly through dark field illumination, we go for culture where we enrich the sample by peptone water telluride, and we incubate the sample for 4 to 6 hours at 37 degrees Celsius, and then we inoculate the loop full of this sample from PWT to bile salt agar, which is at a pH of 8.6. Okay, and then we screen this sample from this bile salt agar into oblique illumination. Now, characterization of Vibrio can be done by many ways. Like when we see the gram stain, we see specific colonies that are translucent, moist, raised, 1 mm around in diameter, smooth, and we see gram negative curved rods. Again, when we do the serology, okay, so while using the specific anti-Vibrio serum, we may see the agglutination. The characteristic biochemical reaction in the case of Vibrio is cholera red reaction, which is due to nitroso-indole compound. And one more characteristic is the Vibrio do not form acid with arabinose. For diagnosis of specific serotypes like Ogawa, Inaba, and Hikojima, we use slide agglutination. So this is what is the summary of lab diagnosis of cholera. Thanks for watching.